Hello, in this video, we're going to talk about gene transfer in microbes. So, microorganisms have these amazing abilities to share genes with each other, and that's what we're going to be talking about in this video. So, horizontal gene transfer is the concept of bacteria sharing genes with each other. And that is different from vertical gene transfer, which is passing your genes from parent to offspring. So, vertical meaning from up to down. That's the normal way that genes are transferred. But bacteria have the ability to basically share genes amongst each other, and that is called horizontal gene transfer. And there are three types of horizontal gene transfer: transformation, transduction, and conjugation. Transformation is really simple. Bacteria just take in DNA that is free floating. Some of these pieces of DNA might have a gene that could be useful for them for survival, whatever. And so they have this ability to just take in free floating DNA, which is really cool. In the next two slides, we're going to talk about the two other types of gene transfer. So transduction is when genes are being transferred through bacteriophages. So as a reminder, bacteriophages are viruses that infect bacteria. And so if a phage infects a bacteria, it can go through the lysogenic cycle where it integrates its DNA into the bacteria's DNA. Now, in this part of the cycle, it will need to eventually remove its DNA out so that it can go back into the lytic cycle. Now, if it accidentally removes a little bit of the bacteria's DNA and then packages it into the new viruses, now this bacterial DNA is packaged inside of a phage. And if this phage infects a new bacteria, it can introduce the original bacteria's DNA into a new bacteria. And so this is another way that DNA can be transferred from one bacteria to another through a phage, basically accidentally picking up a bacteria's DNA and transferring it to another bacteria. And then finally, conjugation is how bacteria can use a pili to share plasmids with each other. So remember that a plasmid is a small circular piece of DNA. Bacteria that have this plasmid called the F plasmid or fertility plasmid are considered F positive. And what they'll do is form a pilus with another bacteria that doesn't have the F plasmid. And essentially, they'll just copy their F plasmid over to the other bacteria. And at the end, now they both have the F plasmid. So not only are they giving their DNA to another, but they get to keep their own copy. So that's an even more amazing way to share genes because now both of them have, those genes, whatever genes there are in this plasmid that could be helpful for survival. And so those are the three main ways that bacteria share their DNA with each other. Hopefully this was interesting, and I'll see you in the next video.